drawing in the presentation. I thank you for accepting to do that. Yeah, so I was at the area of completing the case investigation form and the value for it. We have been under heat to explain why we do not meet the turnaround time. We cannot explain that until we have documented those key times: the date of sample collection, the time of sample collection, the specimen type that you have collected. These are very, very, very important for us to be able to complete and monitor turnaround time. The specimen collection containers. And again, I request colleagues, everyone, every DLFP in every district, by now, you should have had a pool somewhere you have kept to collect at least 10 samples. You should be having about 20 coveralls. You should be having about 10 pairs of gumboots. You should be having about at least one case investigation booklet. You should be having a fine tip marker pen. Every district should be able to have at least those specimen collection supplies. And if you are unable to have, or if you do not have, please ensure you reach out to CPHL, reach out to Rebecca Nachide, reach out to the hub system. They will be able to ensure that every district has these supplies. So the best specimen of choice is whole blood, and we are going to use the EDTA tube, which is this one on this side, the purple top. The second best choice, Winnie Morgan, please help me mute those people. Winnie, help me mute those people. Thank you. The second best choice of specimen to be collected is the swab, and we are going to use the oropharyngeal swab. After you have collected about 2 swabs in the different spots of the buccal cavity, you insert it in the virus transport medium, and that should be able to come through. For breastfeeding mothers, we are able to collect in breast milk, because if somebody has a baby, before we return them to breastfeeding, we must ensure that the breast milk is really sterile and does not have any more particles of the virus. So we are going to collect breast milk and have it tested. And then for people who have survived, who are males, we know that the Ebola virus survives in the testicles between 2 to 6 months to even a year. Studies are also demonstrating that we can reach 1 full year while you're still having that active virus. So you be like a moving bomb. Anywhere you plug, that person will get the disease. So for monitoring such survivors, we use the seminal fluid analysis to monitor these survivors. So these 2 specimens, really, I do not want to hear you running around that you want to collect a breast milk sample from a mother. And this one is going to be used for survivors only, those who have recovered, and we want to see if they are able to go back to breastfeeding. So please do not start saying, I want breast milk containers, I want breast milk containers. Those are only reserved for Ebola treatment units and the survivors program. And also the seminal fluid. Why would you run around telling people to give you seminal fluid just to detect the disease? So that will also not be good for us. So our best tube you should have right now is this purple top. Every DLFP, every health facility laboratory, ensure that you have purple tops nearest to your presence to be able to help us do sample collection. So why are we doing these tests? For diagnostic purposes, for follow-up, a patient is alive, we are going to use nucleic acid amplification testing, the NNAT, and the specimen of choice there is EDTA blood and or plasma. Then for deceased individuals, I had earlier told you, a person who has died, we are going to use swabs. We are not going to pretend to be experts in pathology to be able to collect specimens from hearts of those people who have died. So for us laboratorians, I am requesting you to use swabs to collect from deceased patients. We will bring in board the pathology team, the mortuary attendants, to help with collection of specimens from the heart that will be able to also support this testing. In the meantime, for us laboratorians, let's stick to using the swabs for the buccal or oral area. And we are saying that rub the inner cheek. Rub the swab in the inner cheek of the person without opening the buccal cavity. Okay? Yeah, so then for purposes of surveillance, we might get surveillance request and we want to know if there has been circulating antibodies in a specific community. For example, if we now target and say that most likely this guy or this patient index case has gotten the disease from this district, so we go ahead to do the serosurveillance. That is when we shall use red tops. So again, I'm asking you colleagues, don't run around with a red top and you're saying that I want to use my red top to collect the sample. That one is not acceptable. Plain tubes are for antibody studies, and antibodies are known to appear in blood several days after the infection. So they do not help us in the early confirmation and detection of the disease. Then for patient follow-up, we are going to be able to take other tests, and this is for persons working at the Ebola treatment unit. We're going to be able to support with doing liver function tests, renal function tests, blood grouping for those who will have lost a lot of blood and need the blood for survival for therapeutic means, CSF, urine, and all those others. So again, a team that is very heavily trained will be able to be prepositioned at the Ebola treatment units to support with this kind of sample collection, and small mini labs will be placed in those places because we don't want everybody to analyze these samples in their labs. So they will be restricted to Ebola labs, Ebola treatment units. So cadaver sample collection, I had earlier on alluded to this procedure. You are going to, if you find the mouth already open, the way this picture is showing, you're very lucky. Most times in Africa, when people have died, the first thing they do is close the eyes, close the mouth, tie the head. So if that has happened, do not again go and say, I want to untie the head and open this what? Mouth. The fluid that is building in the body will gush into your face, and you don't want to be in that state of panic. So you just try to insert the swab through any opening within the buccal cavity, and make sure you reach the inner cheek and be able to scrub the inner cheek. Yeah, for the teams that are going to be doing this, we are going to have another training for you, a physical training, to ensure that you are really doing it right in the right way. And then safe sample handling, we are talking of triple packaging. Again, we do not want to expose our transporters of specimens. We are going to go for class A triple packaging, and the primary receptacle is what I had shown you earlier. Whatever receives the sample is actually known as the primary receptacle. And then the secondary, we are going to use a durable, airtight, water leak-proof container. Now, will you know that this container is sufficient? For example, I will give you workable practical solutions. This particular container in this picture is a very expensive container, and you may not have it in your facility, right? So because of that, you go and collect the Septrin or Panadol or those containers that have drugs at the pharmacy. Throw it up and allow it to fall a free fall. When it falls down and does not disintegrate and remains intact the way it is, then it is able to withstand 95% kilopascals of pressure. So that means it is one of the containers you can use. Why do we do that test? Imagine the transporter gets into an accident and something, that fall happens. We don't want that container to disintegrate and spill all the contents within the roadside or in a marketplace. So this is very important that we maintain triple packaging. And then the tertiary containers, as you can see, this cool box. Now we have special cool boxes that we use that also have temperature monitors that are very good to help us support in maintaining this transportation. If you see this procedure down here, this is a quick step in our local setting. Collect the sample, put it in a secondary container. You may not have this tube, but you will have this Ziploc. So from here, you can put it direct into this Ziploc. Before you do that, because you don't have this tube here, you use the gauze to wrap around this container. Now that gauze helps absorb in event of any accident. It absorbs any of that fluid that is found in the floating around. So I would like to stop there and thank you very much for listening to me. And I again insist that specimen transportation is very important, but it should be a safe container that you're handing over to the hub rider, to the hub transporter. Ensure that after packaging, you disinfect all over around the specimen container, the tertiary container. Thank you very much for listening to me, and back to your chair.
Yes, sir. Thank you, Rogers. I think you've been so clear and explicit to your discussion. So, colleagues, one key thing that Rogers brings out is there must always be a request form that must be filled with all the particulars that are required, and we fill in section 4 of the case investigation form. Number 2, he mentions out the clear specimen types, and colleagues, I want us to stick to that. Let's not go to specimen types that haven't been mentioned here, because I know there's always excitement during outbreaks, so people want to take off different sample types, even when there is no need. Someone is a lady, we can take off a blood sample. Someone wants to go and dispense breast milk. Someone wants to take off a semen sample, yet you're a female medical worker. So some of these things, we do not want to see them happen because of excitement or having that need to know some of these other specimen types. Let's stick to what is normal and endeavor that happens. The other thing I want to ensure is that always use universal transport media or viral transport media for the integrity of the specimen to be maintained. And then it's very clear that for the deceased, most times, if you're not an expert who has been trained, like the morticians and the pathologists, do not insist on taking off some of these sample types, like the cardiac blood withdrawal. If you know you have not been trained, please do not insist. Let them do that. So let your roles be very clear. Lastly, that I would want to speak about here that Rogers has made is the triple packaging. We know we do not have the standard materials, but I want to insist that the principle is triple packaging. The primary, the secondary container, and the tertiary containers. And this is just to protect the primary sample, the receptacle sample, where we do have the infectious agent. So we may not have what is really presented here, but we have to practice the principle. He has mentioned about using the available containers that we do have at the facility to guide some of these processes and to ensure that we can attain the triple packaging principle. So at this point in time, allow me to invite Tony. Tony is a laboratory also technologist and a microbiologist as well at the National Reference Microbiology Lab. He has been key in setting up the mobile labs in the country, together with Pimundu, Godfrey, who is also, I think, supporting at NHLDS, and part with also Stephen Balinandi, who have been trained by the East African community in setting up these mobile labs. He has vast experience, and he has been trained in so many molecular techniques, and he's currently supporting, is the manager, the deputy manager of the mobile lab in Mbale. So Tony, I want to welcome you to this section, and then you can take us through. Then we shall have the questions and the other discussions coming in later. Tony, you're most welcome.
Oh, well, thank you, Morgan, for allowing me to speak to laboratorians and possibly other colleagues that are on this call. My name is Tony Muigi. Just like Morgan has stated, I work with the National Health Laboratory and Diagnostic Services and spent quite a lot of time in disease surveillance and outbreak response. I'll briefly take you through the aspect of laboratory reference testing, because it is important for us to know what the reference laboratory testings actually labs are doing, and how you support and fit into the entire process as we are all responding to the outbreak. I request that we go on. Laboratory outbreak response and outbreak confirmation, like the first speaker, that is Winnie Agwang, highlighted, that it is the laboratory that provide confirmation to etiologies that cause public health threats like EVD that we have. So the lab plays a role from identification, confirmation of the outbreak to monitoring of the outbreak. So we are here to support that entire pathway to ensure that we contain this outbreak in the shortest time possible. The laboratory confirmation for also Ebola virus and also Marburg virus is entirely, we are deploying the NATS technology, which is the nucleic acid amplification testing. And that is what is recommended for routine testing and confirmation of Ebola and Marburg in this case. So we have in the country also Ebola virus and also Marburg virus platform, what we call the pan-virus NATs that are able to pick these viruses on real time to fast track the confirmation of these viruses. We employ the molecular techniques of reverse transcription polymerase chain reaction to confirm the two we've talked about. But in our testing pathway, just like my other colleague has stated, so many events present clinically as the two that we've mentioned. So the reference testing pathway takes care of the fact that we shall be doing, and the laboratories are doing differential diagnosis for where suspected individuals that have been suspected for Ebola and Marburg test negative. We subject them to a differential testing for the pathogen we know present clinically as the two. And among those that the reference labs are actually monitoring out, notably that many of us might ignore during the outbreak is the malaria that presents just clinically as Ebola and Marburg. We have the Rift Valley fever that is part of our panel for that we subject the samples that test negative for Marburg and Ebola and CCHF dengue virus that could be circulating. So while we are responding to Ebola in this situation, probably Marburg can be circulating along, CCHF can be circulating. So we are taking account of that and making sure that we don't miss any pathogen that presents as a VHF in this context. We have the next slide that we'll be talking about the labs. I will not talk about the mobile labs here, but we use technologies that PCR platform from Biorad to point of care, biofire film array technologies. There are so many platforms that you use depending on the lab that is running this. But we are very much aware, apart from detecting for the etiology itself, Ebola, Marburg, let's say CCHF, we are aware that when our confirmed cases are taken into the ETU, they are given clinical care. Now, clinical care is supported by other tests apart from the real-time PCR that we offer for the detection. And our laboratories are actually prepared to do complementary laboratory testing, or what we call testing that support clinical care, to include a few that you see there, like for example, the liver and renal functional test, coagulation tests, the blood count, because when somebody is infected with Ebola, we're very much aware the disruption of the entire blood system and the evasion of the immunity. It subjects the organs in our bodies to be very vulnerable, and most of the patients succumb to multi-organ failure. So monitoring the functionality and how to support those organs as care is very critical. And this is is going to happen in the designated labs that we've made mention of. The pathogen we are dealing with is a biosafety level 3, so it carries a weight of a bioterrorism pathogen, and it requires very heightened and improved system for biocontainment that we may not really allow our routine laboratories to do these routine tests, however much they have them in their facilities. So like, for example, for Mbale, the functions of liver function testing, blood count, and so that shall not be done within the routine laboratory that is at Mbale Regional Referral. And this temptation should not be thrown to some of us, for where somebody is suspected to be a VHF and there is a push for you to do some of these tests. This will be exposing you to these pathogens, and what we highly recommend that the sample must be first tested, results issued out, and considered safe for you to run the samples for a person that is known not to be containing any of Ebola, Marburg, or any other VHF. Next slide, please. They have made mention of the labs that so far have been designated to handle the testing of these Marburg, these Ebola-related and VHF suspected samples. And the choice of these labs is informed by the labs selected here to have demonstrated capacity and having validated their assays to perform virus inactivation completely to handle this virus. We use the hazard. These labs have used the hierarchy of mitigation measure to establish ways of how they can eliminate the hazard. The hazard we have here is we are talking about the virus, in which case is Ebola and any other VHF. And the approach we use is that we want to eliminate this hazard using the available infrastructure that give us the engineering control, and supported by our procedures and administrative practices to ensure that we completely eliminate the hazard. Now, the hazard we are talking about here is the virus itself, which is the antigen. And we're very much aware that the biggest problem with that pathogen is a glycoprotein that is transcribed by the GP gene, and that is responsible for entry to our cells and at the same time evading the immune system, and therefore causing the infection that we talk about. So when we remove that glycoprotein and denature it, we render the sample inactive to cause infection. And those labs that have been listed there have demonstrated, and they have been validated, they have been certified nationally, internationally, to completely inactivate, eliminate the hazard of biosafety level 3 pathogens. And so they can handle the subsequent downstream laboratory testing without any fear that there's infection that will happen. So the hazard at that level, which is the active virus with its structure containing the glycoprotein, is eliminated. So the laboratory eliminates that and only works with the nucleic acid, which is RNA. So the labs that I've mentioned there, you have we are talking about tray, and I'm gonna that led to the confirmation of this outbreak. and it is still supporting the testing of samples that are coming within the central, nationally and within the central area of Uganda. We have are doing, including that path of laboratory diagnostics, as it provides valuable information that gives us about the origin of the pathogen that has caused the outbreak. It gives us epidemiological, the data we get from this laboratory helps us to understand the epidemiological and the genomic epidemiology of the pathogen that we are dealing with, and establishing the characteristics of the virus in this case. What is the virulence related and the pathogenicity with this particular strain? So that data is provided by our genomic surveillance, our genomic lab that does that bit. And in doing so, we are able to understand whether this outbreak, this work is still ongoing to establish whether it is a single introduction of this outbreak or multiple spillover events, and probably if there is viral persistence among probably survivors in either human environment or the animal space. So we are deploying that to understand genomically what is highly contributing and what led to the outbreak. So that work is really very underway. Now we have, of course, the Uganda Virus Research Institute that is actually centrally located. It's having also supporting national testing capacity. So it is serving the entire country equally and supporting the outbreak center in actually in Kampala. So you see the capacity that is very rich. And because of we have 2 labs that are centrally located, we have a hotspot that is in Mbale, like it was described in the timelines of events. And we had to deploy the mobile lab testing laboratory at Mbale Regional Referral, which is actually functional now. And purposely is that we want to bring the services closer to the region in Eastern Uganda. One, that is reduce the turnaround time that is contributed to moving the samples to the central area that is Kampala, and of course, providing quality and safe testing within the region. So what you see below, those are the pictures of the labs that we're actually talking about. We didn't, I think the time caught us, we didn't add the photo of the infrastructure we are using in Mbale, but along the way, you'll see it circulating. So you see the Central Emergency Response and Surveillance Laboratory, the Uganda Virus Research Institute that many of us are familiar with, and then the mobile lab infrastructure. So the mobile lab infrastructure is equally either truck mounted or the modular system are BSL-3 infrastructures that are able to handle the material inactivation and holding of the material to provide biosafety and biosecurity that is required in this case. Next slide. So what is our testing algorithm? Somebody would be asking, when do we test and when do we not test? We've been getting these questions, and our colleagues, part of them are health workers and they want to be tested for Ebola, and you get these requests, and these people are not meeting the case definition, and we have a clear case definition for who needs to be tested and who is considered a suspect. That is clearly defined in our technical guidelines, the integrated disease surveillance and response guidelines that describe who meets the definition of a suspect and so qualifies for a test for a VHF. But before I go through this testing algorithm, I want us to pay attention, given that we have a lot of lab people here. And this is where you help us in how we implement this algorithm. My colleague Roger talked about the completeness of the case investigation form. And this is where you'll see where some of those parameters play a very important role. And I want to highlight the section that is very much left out, and that is the onset of symptom. in our testing algorithm, onset of symptom contributes a lot on the decision that is taken on the outcome of the result for the sample that has been taken. So when a person presents with the onset of symptom and meets the case definition, qualifies for a sample collection. And there are two pathways there. The first pathway on your left is if a person has had onset of more than 72 hours from the time they are presented for sample collection. A sample is collected and a reverse transcription PCR is done. There are two likely outcomes. It is either positive or a negative. So for such a person who has the onset of symptom as that more than 72 hours, at that level, it is expected that that person, if it is Ebola and whatever, we have the viral load as established sufficiently that the analyte, which is the agent, is adequate in blood to a level that our testing technologies and pathway are able to capture the target, which is the RNA of the virus. So we are very confident that we are working within the viral peak of that individual. So you cannot miss the target if it is actually there. So when that person is tested positive, is therefore handed over to the treatment, the EVD treatment center for management and care. If that person tests negative, then is handled and then the differential diagnosis for other possible cause of that symptom that qualified them to be suspect for EVD is therefore taken and is managed as a negative for EVD. Now, if somebody has presented the onset for less than 72 hours, at that level, we know that the viral load or the viral burden in blood is considerably low for possibility of being detected by our PCR. So the likelihood still remains the same, either positive or negative. When it is positive, that is quickly regarded as positive and treated. But for such a person, when they test negative, would not want to consider them as negative at that point. We keep them under isolation because we want to collect another sample from this individual 72 hours from that. Why do we want to do that? Is because we want to allow time for, in that period of 72 hours, the viral load and the viral burden has increased in blood that is detectable by our PCR platforms that we have. Now, you see the challenge that we get when we don't get this information and it's not documented on our case investigation. And so decision-making becomes a very complex situation. So we encourage our colleagues here, and usually the lab is the terminal end that handles this case investigation form. We raise our appeal to you to really do the due diligence, support on ensuring that all the parameters are documented by other people who support in taking these cases and classifying them as suspect. They should be able to give us that information as much as possible. So all the sample type that my colleague has talked about, I will not talk about, but that is our testing algorithm. Next slide, please. Yes. We go to the discharge criteria. Morgan, I think you jumped. Yes, please. Now, the discharge criteria is along the way when we get cases into the treatment center. At the end of the day, we have to discharge them upon clearance or we want everybody who gets into the ETU to come out as a survivor. So the earliest we detect them, the more clinical outcome, the better clinical outcome. So the decision on the discharge criteria is driven by clinical outcome. So when we have, on your left, when we have a person who had been in care for 3 or more days without fever or any significant symptom, and significant improvement in the clinical condition, plus ability to perform activities of daily living, that person is considered to be having cleared the virus within the blood. So we start considering them for discharge, but we discharge them after we have established that they have cleared the virus in the blood. So we collect a sample from them and we test. So likely, the likely outcomes are positive, negative. So when the person tests negative, then it's prepared for discharge. When they maintain to be positive, even with good clinical outcome, we do a repeat PCR 48 hours later. If they maintain to be positive, we do another test 48 hours later. But after the first path of 48 hours and they're negative, they follow the negative path. But we have 2 pathways for the lactating and the non-lactating patients. We have mothers that who are lactating, and even if they test negative, we have sites that we consider the privileged sites for immunological invasion, and that is the breast milk, the semen, among others. So our immunity sometimes does not get to those areas to clear the viruses adequately. So people intend to remain with these viruses in those immunologically privileged sites, and the breast milk is part of it. So for the lactating mothers, however much they have tested negative with blood after those sequences of repeat of 8 hours, we actually go further and test their breast milk to determine